And our last speaker before the break is Tyron Moore from Stanford, and he'll talk about the influence of gaze control circuits on visual cortical signals and its relation to perception and cognition.
All right, well, thanks. All right, well, let me start, of course, by saying thanks to the organizers for inviting me to this wonderful symposium. It's great to be a part of it and to be able to tell you a bit about some of the work going on in our lab. All right, so I'm going to start, I'm going to begin this with sort of a bit of a sort of reminder of kind of a basic fact of visual perception, or really any sensory processing. And it's sort of the fact being that there are really rather strict limitations on the capacity of information that we can process at any given time. So for example, and this is pretty easy to demonstrate. So if I have you fix your gaze on one of these, plus either that one or this one, whichever one is closest to you, and if I ask you to hold your gaze there, and I simply present a stimulus in the periphery, you'll find that it's pretty easy to recognize that stimulus as a sequence of flashing letters, in this case an H and a K. However, if I now try two pairs of alternating letters, it's still fairly easy to process. You can tell what the letters are. You can pair them for different locations. However, if I start to escalate things a bit up to say 6 pair, it suddenly gets much harder to identify the sequence of letters each of the locations. At 18 pair, it becomes harder still, and by the time we get to 40 pair, it's totally impossible. Even if the stimulus array is even though it's being presented multiple times, none of you are able to remember, process, and identify all the letters at all the locations. All right, so it's pretty clear that visual perception has a pretty severe limitation on the capacity of information you can process at a given moment in time. And since a typical environment contains more than just a few items, it means that most of the time we miss much of what's right before our eyes. However, one sort of bright spot to all this is that we nonetheless have some control over which items we actually do perceive. So for example, if you know that in that sequence of letters that there's one item or one location that's particularly relevant for your purposes, say for example, if I give a little cue there, you'll find that it's pretty easy to sort of identify that one a series of letters at that location. And you can do it with your gaze still fixed on the spot there, so you can recognize that it's a Q and a T. All right. That is, you can covertly attend to that location and process the relevant information and ignore the distractors, right? So even though your gaze is fixed here, you can still selectively process that location. All right. So over the past few decades, numerous studies have established that the visually driven activity throughout most of the stages in the primate visual system is modulated by the direction of covert attention. And so just as in humans, we can train monkeys to play the similar games that you just did in the demo. That is, we can train them to fix their gaze on a central fixation spot. We can take one of multiple stimuli and flash them on the screen and have them perform tasks. We can even take one of those stimuli and place it in the receptive field of, say, a neuron and visual cortex. This is a side view of the macaque monkey brain here, and this is, I'm showing you the exposed part of area V4. So we can record the responses of neurons in the visual cortex, while an animal has to play the same game that you've played. That is, on some trials, it has to monitor the identity of a stimulus in the receptive field and ignore another one, or the reverse. And what you often get is shown here on the right. This is just the instantaneous spike rate of a neuron recorded in area V4. It's aligned to the moment at which the animal gets a cue as to which stimulus is the relevant one, the one in the receptive field or the one in another location. And what you see here is what you typically get throughout the visual system. That is an increase in the response at the attended location, and no change, or in some cases, a decrease in the response at the ignored location. Now, in addition to changes in firing rate of activity of neurons, there are other changes in the properties of responses that relate in some way to the information that the neurons can convey about the stimulus that's being coded. So, for example, there can be a diminution in the variability across trials, so the increase in the reliability of spiking responses, increases in the tuning, say, for example, to orientation or color or whatever, as well as in some conditions where it's compatible with increases in the information conveyed by populations of neurons, you can get decreases in the correlated variability, so a decrease in the redundancy of responses. All right, so this stuff here, the neural correlates, we pretty well understand. Well, work in my lab over the past several years has focused primarily on trying to identify the source or sources of the modulation of visual signals in general, as well as during attention. That is, we've been interested in identifying the mechanism by which an animal is able to sort of willfully control the gain of visual signals according to behavioral demands. And some of the initial insight into sort of the causal basis of attention, at least visual spatial attention, comes as early as the late 19th century from David Ferrier. Now, David Ferrier, as many of you probably know, was a sort of a pioneering experimental neurologist who used brain stimulation to map out a lot of different sensory motor areas of the cortex. In addition, David Ferrier also did a large number of lesion studies in various animals, monkeys and dogs and so forth. And it was through these lesion studies that he recognized that in addition to to deficits in motor control upon damage to frontal lobe areas, there are also there are these other deficits that are more cognitive. And Ferrier hypothesized that the power of attention was really related to volitional control of the head and the eyes, so gaze control. And he also went even further and seemed to sort of speculate that when attention is directed covertly, that is in the absence of movement, that those movements are suppressed, but the preparation of those movements is still nonetheless affecting sensory factors. So it was a pretty insightful stuff. And so sort of we mapped that hypothesis roughly onto the sort of classic paradigm of covert spatial attention in neurophysiology. We can imagine that one hypothesis would be that if an animal is to attend to a peripheral location, that attention might come in the form of a preparation of a gaze shift to that location. That is, the animal may prepare to move its fovea from this location to that location when it's attending here versus attending there. All right, that is, the preparation of gaze shift to the relevant stimulus may be sufficient to improve the fidelity of visual processing at the relevant stimulus. All right, so consistent with this idea, there's been sort of an abundance of evidence over the past 10 or so years implicating oculomotor structures in the control of visual spatial attention. In particular, there's now very good evidence of a causal role of the frontal eye field in attentional control. And the FEF is just shown here in the anterior bank of the arcuate sulcus. This, again, is a side view of the macaque monkey brain. And the FEF is essentially the part of prefrontal cortex that's most directly involved in controlling eye movements. And this involvement in the control of saccadic eye movements is achieved by way of the FEF's output to the superior colliculus and the brainstem nuclei, and eventually out to the eye muscles. But in addition, the FEF is also heavily interconnected with most of the areas in extrastriate visual cortex. So beyond area V1, FEF neurons receive input from visual cortex, but also send inputs back to visual cortex. And that latter top-down projection might be the basis by which oculomotor commands might shape the responses in visual cortex. All right, so thus far, and after to sort of summarize very, very quickly a lot of work, we pretty much know that by activating neurons within the FEF, that's sufficient to bring about the deployment of spatial attention to a particular location in space. And at the same time, it's also possible to bring about the modulation of visual responses within visual cortex with that same activation. We also know that the FEF appears to be necessary for normal attentional control. If we inactivate it, then animals will exhibit attention deficits. But what we don't know, however, is the extent to which signals that come from the FEF that are related to the preparation of eye movements and the signals that are used to modulate attentional deployment and modulate visual responses and visual cortex are emanating from the same groups of neurons within the FEF. The FEF has consists of a variety of different functional classes of neurons and different signals. And so I'm going to tell one little story today about how we've begun addressing the extent to which those two signals emanate from the same neurons or the same neural types. All right, and so there's sort of two kind of possible competing hypotheses. So as I alluded to, the frontal eye field has different classes of neurons. There are neurons that look pretty much like what you see in posterior visual cortex. They're just purely visual cells. They have receptive fields. They respond to visual stimuli. And there are neurons that have movement bursts or movement activity related to saccadic commands. And there are many cells that have both properties. And one competing hypothesis has it that it's really just the visual cells that we know are modulated during the deployment of attention, just like you see in visual cortex, are the only ones that are actually projecting back to visual cortex, whereas the ones that are involved primarily in motor control, the ones that project out to the colliculus and the brainstem. And so this would be sort of the case in which the two populations really aren't the two signals aren't being mixed, and more importantly, that information about the preparation of gaze commands is not conveyed back to visual cortex. And then alternatively, another hypothesis, another possibility that's quite different is that there are actual signals related to movement commands that are conveyed back to visual cortex, right? So these are very, very different possibilities, and we don't know, there really isn't any clear evidence up till now about which might be true. So I'm going to tell you about an experiment which we tried to, one of the experiments in which we tried to investigate, to isolate which one of these is in fact the case. And it's really just the work of a former graduate student, Nick Steinmetz, who sort of posed the question, does saccade preparation modulate visual cortical responses even when that saccade preparation is dissociated spatially from attentional control? So if we go back to our hypotheses here, if indeed this is true, this means that if an animal is preparing an eye movement to point A, but attending to point B, then the modulation you'll see in visual cortex will only correspond to point B. You'll see no signals really regarding the preparation of saccades to point B. So a very simple design, or a very simple question. If, however, there are motor signals that are sent back to visual cortex, then the preparation of a saccadic eye movement should be sufficient to modulate activity, even though there's no attentional demand. All right. So, and the way in which Nick addressed this question was by training animals to, on a task that's very similar to the demo that you performed earlier. That is, it's a task in which the animal maintained fixation spot and had to identify changes in a stimulus in the periphery across a flash presentation. Now, you probably didn't notice that that one of these oriented gratings actually changed orientation across the flash presentation. You didn't notice it because the flash itself introduces a transient that masks that orientation change. This is a classic change blindness. If I get rid of the flash in between the two sets of gratings, you'll easily spot a change in orientation, right? That just pops right out. I don't know if all of you are paying attention, but so to speak. Well, it actually doesn't really require much attention without the flash. But however, if I give you a cue as to which one is likely to change, so if you keep your gaze fixed there, and I bring back that flash presentation, you'll find that it's easy to see the change in orientation, right? So you can overcome that masking of the flash by simply attending. So we had monkeys perform this task. They're quite good at it. But there was an important twist here. So I'll walk through the task design. So first, the animal achieves fixation, and during this task, we're going to be recording from sets of neurons recorded more or less in the same columns of area before. So there'll be a bunch of receptive fields that'll be positioned somewhere on the display. After fixation, a set of 4 gratings would appear, one of which appears in the receptive fields. And then after another delay, a cue that tells the animal, a cue appears at the fixation spot. It's much smaller than this, but I made it larger so you could see it. It's much small. It's quite small. That indicates the direction at which, the location at which the grating might change. And so, and I'll get to the numbers in a second. But and following that cue, the animal, of course, that cue tells the animal to attend at that location. Then there's a blank, then the array comes back on, and across the array, the orientation at the cued location might change, as it does here. or it might not change. It might not change. If it does change, the animal has to shift his gaze to the opposite location. All right. So that's the animal's gaze. So if it changed here, saccade, make an anti-saccade to the opposite location. Right. If it doesn't change, then maintain fixation. These are catch trials. So if it doesn't change orientation, maintain fixation. Now the cue was almost 100% valid, meaning that if the cue indicated up and to the right, in this case, that on fewer than 5% of the time across many experimental days, did the change, if it happened, occur at another location. So it's a very reliable cue. But the change only happened on half the trial. So the animal knows, once the cue appears, the animal knows that if there's a change going to happen, it's going to happen at that location. Right. But the animal knows another thing. He also knows that once the cue comes on, he also knows the most likely location to which he has to move his eyes. Right. So if there is a change in half the trials, the saccade will have to go to this location. If this is cue, then the saccade will go to this location. All right. So in this way, we're sort of dissociating the deployment of spatial attention to these gratings and the preparation of saccades right. So let me summarize again the trial conditions because it's quite important. So again, the sort of classical if you will, condition is when the cue occurs at the receptive field. That is the case in which we expect the animal's attention to be deployed at the receptive field location. Then there's the cue opposite location when the cue indicates that the opposite location is the location of attention, and therefore the receptive field stimulus is the most likely saccadic target. Okay, and then the other conditions are basically neutral. They're orthogonal to the saccade plan and to the attentional deployment. So where the cue either happens here or happens there. Okay, and we're going to add these guys together and use them as controls. Right, these are the case in which the animal is neither preparing a saccade nor is it attending to the receptive field stimulus. All right, so first let me walk you through a bit of the behavioral data to convince you that the task is actually doing more or less what we want it to do. So first of all, this is sort of the animal's performance in detecting the change across that flash when the cue is valid. That is to say, if I cue to the left and it actually happens at the left, what's the animal's performance? And so when it's valid, the animal performs very, very well. And on those rare trials in which we surprise the animal and do it at some other location, the performance is quite poor. And it doesn't matter if it's at the opposite location or if it's at the orthogonal location, right? So the animal's attending, by this measure, is attending at one location, which is the cued location. Does that make sense? Okay, see a couple of confused looks right there. I'll come back to you, I guess. All right. Another sort of classic measure of attentional deployment is the reaction time, the speed at which the animal actually renders a correct decision. And so again, when the cue is valid, the animal responds with an opposite saccade to a location. That the reaction time is much faster than when the animal responds following to a change at an unexpected location. Right? It's a bit higher for the opposite location, and intermediate for the invalid location. So this is the usual sort of classic evidence that the animal is attending to really just the valid location following the cue. So in that post-cue period. Okay, so we got the attention part down. What about the saccade preparation? How do we know that the animal is using the information about the trial structure to plan a saccadic eye movement at the most likely location? So before the cue, the animal, it's a 1 in 4 chance, no, actually a 1 in 8 chance that the animal will have to make a saccade to either one of the 4 targets. Once the cue occurs, it's 1 in 2, right? Because once I cue this location here, he knows that half the time he'll have to move his eyes there. So, and so the way in which we demonstrated to ourselves that the animal actually was aware of that information was by looking at the trials in which the animal broke fixation and aborted to made an abortive saccade to one of the gratings. So this is one such case where this was the cued location, and these are a set of trials out of many hundreds in which the animal broke fixation and saccaded to the opposite location. So so just by the numbers and by definition, the animal was more prepared to saccade to the opposite location following the cue, right? So if we take that one example, we sort of look at the sort of all the trials from across 2 of the animals, we see that the animal is much more likely to break fixation and saccade to the opposite location. So by definition, the animal is preparing a saccade in one direction and attending to the other direction. So based on these data, all right. We'll probably return to these figures at the end. But anyway, all right. So okay. So now we have the behavior more or less in hand. We want to ask our main question, which is to what extent does the Saccade preparation modulate responses? Okay. So these are three example V4 neurons recorded on different days, probably. And so this is kind of the classical result. That is, if you the sort of light blue is the case in which the animal is cued into the receptive field stimulus, right? So in each case, following the cue, once the animal knows where the likely change will be, the responses to that stimulus are enhanced, that we fully expected, whereas the responses to stimuli in the orthogonal trials, that is when the stimulus in the receptive field is not the target stimulus, the cued stimulus, and the responses often either stay the same or drop a little bit. So this we fully expected. Now, but the real question is, what happens in the case when the animal is attending opposite the receptive field, but is preparing a saccade into the receptive field more than it does on the other trials, right? So again, if there's no information about the preparation as distinct from attention, then we should expect these trial conditions to look just like the orthogonal conditions. If, however, there's the preparation is sufficient to modulate responses, we should see effects just like these, and that's exactly what we see. So even though the animal is preparing a saccade to what is effectively a distractor to the task, the visually driven responses in V4 are modulated just like they are for the attended case, right? In fact, this is very representative. So if we take the responses of a full population of neurons, this is both single neurons and multi-units, I believe, from 2 monkeys, and this is just we're making an index here, as one often does. So we're just comparing the case in which the animal attends to the receptive field stimulus minus when the animal when the cue is to one of the orthogonal stimuli, and it's divided by the sum of the two, and so a rightward shift would indicate that the responses tend to be greater in the attention case. So this we fully expected again. Now the question is, what happens in the saccade case? Well, in the saccade case, it's the same. In fact, the rightward shift of this index is actually larger than this index. So that is to say, the effect of preparing a saccade to a distractor is actually larger than the effect of attending to the cued location. All right. And actually, comparing this modulation, which is something around like I think 4 or 5%, it's actually quite a bit small for V4. 4 or 5% modulation is more like V2 or maybe even V1. So and I'll come back to that as to why that might be smaller than what one normally gets. But in any case, the effects of saccade preparation are larger. Modulation of the firing rate are larger than the attention to the receptive field. All right. So what about? So while we did this experiment, we also varied the orientation of visual stimuli. V4 neurons are orientation selective. And we know that spatial attention modulates the tuning properties of V4 neurons. That is to say, the tuning sharpness, if you will, or at least the gain of the tuning function is increased with attention. So we could take our orientation tuning functions and we could fit them with von Mises function. And what I'm going to do here is just going to show you some examples of these functions. But I'm going to summarize the tuning measurement as simply the tuning amplitude. That is to say, the peak response versus the trough response. So just the peak versus the preferred versus the non-preferred response as the amplitude. And I'm going to compute an index from that, where the peak versus the trough for the attended case minus the peak versus the trough for the orthogonal case, right? And divided by the sum of the two. All right. So we already know that it's been shown before by John Mounsell and colleagues that the spatial attention increases the tuning amplitude. So the difference between optimal orientations and suboptimal orientations increases. All right. So we got this here. Now the question is, what happens when the receptive field stimulus is actually the stimulus the animal is preparing a saccade to, but as a distractor? I mean, the change in orientation at the distractor location is actually distracting. It's not actually important for the task. Nevertheless, we see the same increment in tuning amplitude across neurons. So this is just for one example. The tuning amplitude modulation is a little bit smaller in this example, but here's an example where it's actually larger. That is to say, so the orientation tuning is actually sharpened by a saccade preparation. All right, so we're looking across the population of neurons for the cue receptive field condition when the animal's attending to the receptive field stimulus. The increment in tuning amplitude is about 9%, which is about typical. However, for the saccade case, it's also about 10%, actually a little bit greater. So again, the preparation of a saccade to a distracting stimulus is sufficient to increment the orientation tuning, in spite of the fact that it's not ideal for the task. All right, so in addition, we looked at other known effects of spatial attention. For example, the change in low frequency and LFPs are recorded at the same sites we recorded the neurons from. So it's well known that the gamma band LFPs are incremented during spatial attention, and that the lower bands are actually suppressed. And this is showing you for the attention case. Well, we found that more or less the same was true for the saccade case. So the preparation of a saccade was also sufficient to modulate the LFPs in the same way that we know that they are modulated during spatial attention. All right, and so one last result that's a bit newer is that while doing these recordings, we recorded the activity from before with these laminar electrodes, which I love, by the way. And we did this in part to be able to record from neurons that were distributed across the laminae within their AV4. Well, of course, in monkey cortex, the cortex is folded up, so we had to use the MRI images in order to sort of plot out the contours of the gray matter within the recording well, and then adjust the angle of this laminar electrode accordingly to be able to record from neurons that have overlapping receptive fields. So if you're in the same column, then by definition, almost you will have receptive fields that are concentric across the laminae. All right. So we were able to achieve this in many recordings. And so we wanted to ask, well, number one, does modulation of visual cortical responses, V4 responses, vary as a function of laminae? But also, does it vary between these two behavioral conditions? And the answer to the sort of quick answer to both is no. That is, the firing rate modulation, and this is just showing on the y-axis, I'm showing you sort of a position normalized plot of laminae. It's not really layers. It's the middle channel of a laminar electrode that is positioned within the cortex, and the penetration is normal to the surface, and the receptive fields are all aligned. So this is roughly near layer 4-ish. But the point is, and these are all compared with its own control. But the point is that the firing rate modulation doesn't seem to vary as a function of laminae, either for the attentional case or for the saccade case. That we can't. That there's no distinction between the two. All right, and so those data from that experiment suggest to us that we know that the preparation of saccades is sufficient to modulate responses in visual cortex. So we know that those neurons down there in the ventral stream are actually getting some input from neurons that know about the upcoming saccade plans. And so we thus far favor this conclusion, that is to say that neurons that convey some information about motor preparation are feeding back directly onto to V4. And I don't have enough time to. If I could maybe squeeze in a few, maybe not. I'll get questions. I'll do it in the question and answer. Okay, so let me summarize here then. All right, so the preparation of saccadic eye movements is sufficient to modulate the visual responses of area V4 neurons, even when the saccade plane is spatially dissociated from an attended location. That is, even when that location is irrelevant to the task at hand, or to the attentional deployment. The modulation includes increases in orientation tuning at the saccade goal, resulting in an equally enhanced orientation discriminability of visual stimuli at the attended and saccade target. And the modulation of local field potentials also appears to be equal between the two behaviors. And briefly, the magnitude of modulation by tension saccade preparation is approximately equal across the depths of the cortex. And one conclusion would be that neurons conveying signals related to Saccade preparation appear to modulate visual cortical responses regardless of attentional demands. That is, again, it's sufficient to prepare a saccade. And these movement-related signals likely contribute to the overall gain modulation of visual cortical signals when attention and saccade plans are actually directed to the same location. And that brings us back to the point I made earlier about the attentional modulation being unusually small for V4. And so we think that that's partly because there's one resource that's being, that over the course of many months of training, the animal is basically taking one mechanism, if you will, and it's being cut in half, partly for the attentional demands and partly for the motor demands. All right, well, let me thank, again, my graduate student, Nick Steinmetz, who's now at UCL, and other members of my lab, as well as the support. Thank you.
So if you're seeing similar modulation in the visual cortex in terms of, firing rate, reliability, tuning, why don't you see the behavior improvement?
Yeah, that's a tough one. That's a great question, though. Yeah. So there's kind of two answers, and they're both complicated. All right. So the least complicated answer, I suppose, is that, again, I kind of sort of don't like experiments like these because any time in which you have an animal doing something that's not normal and has to learn that over the course of many months, you don't know what you have in the system later on, right? So in the human literature, there's a set of studies that have tried to test whether or not it's possible to dissociate the perceptual benefits of attention from saccade plans. And most of the better studies indicate that that's a no, that if you come in off the street and you do this, you do a paradigm in which you have to make a saccade and also attend to stimuli, generally you're better where you're going to saccade to, regardless of where you're trying to attend to. There are one or two that dispute that you actually can do that. And so, okay, well then maybe though after training for 9 months, maybe you could do that, and who knows what that means. Secondly, I remember I showed you the behavioral data that animals were better at percent correct at hits of changes at the valid location and much better than the invalid location. Well, obviously, what we really want for that is a D prime measurement for whether or not the sensitivity to the change actually is highest at that location. Well, this paradigm is as you probably know, this paradigm we use because it's a multi-alternative response task. It's very difficult to measure D primes in this paradigm to factor out the bias. But you can see how it would be a problem because in this case the response bias is part of the experiment. That is to say, when you attend here, your response to move to this location versus that location is very different, right? So one of our collaborators basically invented a model to be able to extract D primes from multi-alternative response tasks and use it to fit various data sets as well as this data set. And for at least one of the monkeys, it fit pretty well and suggested that the sensitivity as dissociated from the criterion was actually a little bit elevated at both the saccade location and the attended location. So if you define attention as a change in performance of the neurons, then definitely there is attentional effects at both sites, right? But it's measured behaviorally. So it is possible that there is some benefit at both locations, but in a way that's kind of part of the experiment. It's like can you if the basis for that modulation and response to the modulation and the behavior originates from an oculomotor plan, they should be impossible to dissociate regardless of the task you train the animal on. So there's no incentive of the animal whatsoever to attend at the saccade location. So that's the complicated answer. Right?
One more question?
Yep.
So the experiment The experiment is based on the idea that one of the locations is actually attended, and the other one is a saccade target. But could it be that the animal is attending to one to detect the change and attending to the other because it is the stimulus that is cueing the where he's going to saccade, in which case you would expect.
Yeah, right. So I mean, again, if those two functions are separable, there's no reason why there should be any perceptual benefits at the saccade target, right? A priori, there's no reason. It's not useful to the animal. It's not adaptive. It's not going to increase the.
It is, because it's actually going to saccade there, so it wants to see where the stimulus that is cueing its saccade target is, so that it can actually.
If there is a perceptual benefit, you can explain it in terms of that maybe it's beneficial. But remember, one of the hypotheses is that these two things are separable, that you can prepare an eye movement to location without any attentional benefit. So if that is one of the hypotheses, and so if that's true, then it should be possible to have separable benefits, one at the attention location, and that's improved above that of the saccade location. But they're not separable. I think if we accept the kind of marginal, they're not marginal, but if we accept the data from the one monkey that did fit the multi-alternative D prime model, then essentially, like as I said before, there was both a behavioral experiment, which is it possible for an animal over many months to learn how to dissociate these two demands, and then if we accept that, we'd say the answer is no, they can't, and so. Yeah. Anyway, but I mean, that in the context of the other evidence that we have, it's pretty clear that the signals, at least from the FEF, that are getting back to extrastriate cortex, contain a lot of information about prepared saccades, and that probably is the basis for the modulation back there. I saw. Oh, okay. We're out of time.
Actually, maybe we should head towards the break. You can talk to Tyrion during the break. Thank you.